Welcome, grade 10s. This is week 10 of Cell School for Life Sciences. In our lesson today, we're going to be looking at the differences between mitosis in plant and animal cells. You have learned that cells need to divide in order for a multicellular organism to grow. And of course, new cells also need to be produced so that cells that have become damaged or old can be replaced. And so mitosis is the process whereby ordinary body cells or somatic cells, let's remind ourselves of that word, somatic cells, normal body cells, either in the plant or in the animal, are able to divide in order to produce identical daughter cells. So the process is basically the same as you have seen when you looked at the different stages of mitosis last week. But there are some differences that exist between mitosis in plant and animal cells, and that's what we're going to look at today. Here are some photographs that show mitosis happening. Can you identify the different stages? For me, this is the easiest stage to identify. This is metaphase. And why is it so easy to identify? Well, all of the chromosomes are lined up along the equator. This stage is also fairly simple or fairly easy to identify. This is anaphase. And of course, you can identify anaphase because the new chromosomes, the daughter chromosomes, are moving towards the pole. These chromosomes are moving to the top pole of the cell, and these chromosomes are moving to the bottom pole of that particular cell. In this diagram, or photograph, I would like you to focus on what is happening, what we can identify here. We can see that we have a complement of nuclear material and chromosomes at the top of the cell and at the bottom of the cell, and this is the structure that I want you to focus on. This is known as a cell plate, and this is an indication that this mitosis is happening in a plant cell. Of course, you could have said, but I knew that straight away because the plant cell has a rigid cell wall, and in all of these photographs you can see evidence of this rigid cell wall going around the outside of the cell, and of course that is what helps to keep the cell in shape and helps to support the cell and in fact the plant as a whole. But here in telophase, you can see that a cell plate is being laid down to divide the two cells. Let's have a look at that in a different photograph and see what is happening. This is another photograph showing a plant cell, and in this plant cell we can see that we're in telophase once again, and here you're able to see the two complements of chromosomes. We can even, if you look carefully, see the remnants of the spindle fibers. And at each pole of the cell we can see the new daughter chromosomes becoming the new nucleus. The chromosomes are no longer condensed, they're going back to their network phase. But it is this structure here that I need you to focus on. This is the beginning of the development of a cell plate. Now, what happens is in the middle of the cell that is starting to undergo cytokinesis, cellulose will start to be laid down and will spread out in all three dimensions to finally seal off the two cells from each other. And that cell plate, the cellulose, remember that cellulose is the substance that makes up the cell wall, that cellulose cell plate is a clear indication that you are looking at a plant cell undergoing mitosis. Of course, also very clear in this photograph is the rigid cellulose cell wall that goes all the way around the cell. Now, of course, an animal cell doesn't have a cellulose cell wall. What you're looking at here are two animal cells. Here is the cell membrane, and here is the cell membrane of the original cell. We can see that this is right at the end of anaphase. The chromosomes don't quite have a proper nuclear membrane around them. We can see the little remnants of the spindle fiber, but what we can also see is this constriction of the cell membrane, and the membrane will constrict further and further until we have separated two cells from the original parent cell. And so in an animal cell, we can notice that there is a cell membrane only, which is not rigid, and we can see that the cell membrane constricts. This means it pulls tighter and tighter in, and of course this movement of the cell membrane is controlled by microtubules. So one difference between a plant and an animal cell undergoing mitosis is how cytokinesis will take place. Plant cell, laying down of a cell wall through a cell plate. In the animal cell, membrane constriction. There is a further difference between plant and animal cell mitosis. Let's have a look at that now. In animal cells, we find what is called a centrosome. We find something similar in plant cells, but in animal cells we see that the centrosome is made up of two sets or two centrioles, and these are not present in higher plant cells. By higher plants, we mean the flowering plants, for example. The centrioles, as you can see, are made up of a very specific arrangement of microtubules. And these microtubules are at right angles to each other. And this here is an example of a photograph, a micrograph, electron micrograph, that show, I think I need to use a different color pen, here we go, that show one set of microtubules, and then at right angles to that we see the other set of microtubules. And these are the two, there's one of them and there's the other one, centrioles. And it's the two centrioles that actually produce spindle fibers. They produce the spindle fibers which attach from the poles, they form the shape of the spindle, and we find that the chromosomes lie along the equator of the cell, remember, and they're duplicated, so two chromatids, and they are attached to these spindle fibers. And it's the spindle fibers that move, the microtubules pull apart the two chromatids by splitting the centromere. Now, I said to you that in animal cells we have centrioles. In plant cells, there is a centrosome that automatically produces the microtubules that make up the spindle fibers, but there are no centrioles. Here is your homework question. How would you recognize plant cell mitosis from a picture or a photograph? Remember that the first thing that you would notice if you had a really high-powered electron micrograph is that in a plant cell there are no centrioles. Secondly, even with not such a high-powered microscope, you can see it very clearly that the cell plate divides the two cells. The cell plate is laid down between the two cells during telophase and cytokinesis to separate the two cells from each other. That brings us to the end of our lesson today on the differences between plant and animal cell mitosis. I hope you'll join me again for our lesson tomorrow. Goodbye.